All right, we are at 4:01, so let's get started. Thank you for joining us for the Foothill College COVID vaccine education series presented by the STEM Division and Science Learning Institute. My name is Ram Subramaniam. I serve as the Dean of the STEM Division at Foothill College. This is a four-part series to learn about different aspects of the COVID vaccine. Different aspects such as biology, public health information, myths about the vaccine, and also some information from our Santa Clara County Health Office. Sophia Kim, Rebecca Ryan, Diane Denagel, and Sara Cooper are the co-organizers of this program. I am now handing over to Sophia Kim, who will go over some logistics regarding this webinar.
Thanks, Ram. Hi, everybody. My name is Sophia Kim. I'm the director of the Science Learning Institute at Foothill College, one of the co-hosts of today. Just to review some logistics, as you'll notice, we've disabled the chat box. That's just to ensure that we have full focus and participation focused at our speaker. We would encourage you to use the Q&A box. You'll see it at the bottom of your screen, which you can kind of turn on and off at any time. Please feel free to add your questions as they come. You'll also notice that there's an option to be able to upvote any question that you see if you don't want to repeat another question that has been asked. This is a note that we are recording this webinar and we'll be posting that to the COVID Education Series website, which is where you hopefully found today's Zoom link. So that should be posted in the next day or so. And finally, you will see we've got two of our American Sign Language interpreters with us today. So we've got their screens pinned. You'll be seeing them on screen as well as our speaker. And I'll pass it back to you, Ram.
Thank you, Sophia. All right. So it is with great pleasure and honor that I introduce the first speaker of this education series. Speaking to us about the biology of the virus and the vaccine is Dr. Sara Cooper. Dr. Cooper is a microbiology instructor at Foothill College, and Dr. Cooper's undergraduate education began at the University of Colorado, following which she did her doctoral work at Stanford University, where she worked on Cellular and Molecular Biology. And Dr. Cooper has recently been teaching an honors class about COVID-19 at Foothill College. Regularly, she always teaches our microbiology class offerings as well at Foothill College. Outside of her role as an educator, Dr. Sara Cooper is a parent, a partner, and an avid runner. Please join me in welcoming Dr. Sara Cooper. The floor is yours, Sara.
Thank you. Hi, everybody, and welcome to the first of our four-part series about COVID-19 vaccines. I'm really excited to have you here. I am pretty passionate about this topic, as you'll find out. I think the science is super interesting, but there's also a lot of other really important things that we'll talk about today, too. I was actually just thinking kind of in the last week or so as I was preparing this talk about kind of the difference between a year ago or a little more than a year ago and today, and thinking about how, back in winter quarter of 2020, I was in the classroom with my students. We were in the lab growing bacteria, and we were actually following pretty closely the emergence of this virus as it came out of China, watching it kind of spread, starting to get a little uncomfortable with how it was spreading, and it was a very interesting lesson in microbiology as I was teaching a microbiology class, and there was a lot of stuff going on in my personal life too. My daughter and I had just gone to New York City in February, the end of February, to see a bunch of Broadway plays, and my son was on a school trip in South America. My daughter was getting ready to go to China on a school trip in May. And all of I was planning on going to Costa Rica for spring break. And all of this was happening in the context of kind of this virus spreading and us getting more and more worried about it. And so I just want to keep in context, like I'm about to talk about some big numbers, but remember that this is a really specific event, a really individual event too. Things changed at the drop of a hat. We were online, you were taking classes online, I was teaching online, I was watching students lose their jobs and get sick. I was watching them struggle through sick family members, and in the context of everything else that was going on around us, it was a really hard time, and it still is a hard time. I think the hope is that we see a light at the end of the tunnel with these vaccines. And so today I really want to focus in on the biology of the virus and how understanding that biology has helped us to develop these vaccines, which are going to get us back to a more normal life and hopefully get me to Costa Rica at some point, since I had to cancel that trip. All right. So we're going to start just by looking at the impact of COVID-19. It's a really individual struggle that we've all had in the last year, but it is also an international struggle. More than 141 million people have been infected with this virus and have been sick. More than 3 million people have died. I'm just going to pause there because 3 million people is a huge milestone, right? In this country, almost 32 million people have been sick, some of them severely sick, hospitalized, with long-term effects that they will be dealing with for months, if not years from now. We've had 566,000, more than 566,000 Americans die from COVID-19 in just over a year. It's been a dramatic change in our lifestyles. COVID-19 is now the third leading cause of death between heart disease and cancer in this country. These are big impacts. We think the International Monetary Fund says that we've lost more than $22 trillion from our global economy, okay? A really big thing that's happened caused by a really tiny virus, super tiny virus. This is a cartoon of SARS-CoV-2 that I stole from the New York Times. The virus itself is only about 100 nanometers across. And it's really actually hard to tell somebody how small 100 nanometers is. It is 1 billionth of a meter, right? This virus is about 1/1000th of the width of a piece of hair, if you can think about that. There are more than 10 trillion viruses that would fit into the size of just a drop of water. So it's a really tiny virus. In fact, I have a mathematician friend who did some math and calculated out that the amount of SARS-CoV-2, the virus that causes COVID-19, that exists in the world today would fit in a soda can. So taking it from all of the sick people in the world and putting it into a soda can, it would all fit. So it's super tiny virus that's causing all of these huge impacts on our lives and on our communities. And today we're going to really focus in on the biology of it. So the virus itself is actually called SARS-CoV-2. This stands for Severe Acute Respiratory Syndrome, Coronavirus 2. Okay, the disease that's caused by this virus is called COVID-19. So we're going to talk about the virus first, and then we'll talk about the disease. The virus itself, like I said, is super tiny and actually really super simple. It really only has about 3 components to it. It has a genome made of RNA, and this genome contains all of the information you need to build a virus. So all of the kind of genetic information to build a virus. That genome is encapsulated by a combination of an envelope and a capsid, which are made of proteins and fats. It's really just kind of this tiny little oily blob. And sticking out of the tiny little oily blob are these really important proteins called spike proteins, and we'll be talking about them quite a bit today. That's it. That's this virus. It's not actually something that's able to replicate or really do anything on its own. It needs the help of a cell, one of your cells, in order to do anything, in order to really carry out its effects. And so if we were to talk about how that infection happens, we actually need to not only understand the structure of the virus, but also we need to understand the structure of the cells it's infecting. So you have lots of cells in your body. Many of them actually have a protein on the surface of them called ACE2. This protein is critical for the infection by SARS-CoV-2. The spike proteins on the virus actually bind directly to ACE2. And when that happens, the virus is able to bind and then travel inside of that cell. So the spike binds to ACE2 and enters the cell. And it's at this point when it can start to use all of the components and building blocks inside the cell to start to replicate. So this virus will actually make copies of itself, and it has to copy all of the different components that we just talked about. So it has to copy the RNA genome. It has to copy those proteins and fats that are in the capsid, and it needs to copy those spike proteins that are sticking out of the surface. All of this viral copying ends up damaging the cell and preparing the cell so that these viruses then can escape from the cell. They're going to leave the cell that they were infecting, many more of the viruses than first entered the cell, and they'll be able to go on now and bind to another cell that also has the ACE2 receptor, get inside of that cell, and continue the process by replicating inside that cell. Okay, this process continues on and on and on until you have a whole mess of infected cells. Those infected cells are sitting right next to other cells that don't have infection, but these viruses that are escaping are able to bind, enter the cell, replicate, so on and so forth. Okay, so this is how an infection gets started inside of your body. The question is, how do we stop it? So your immune system actually has a well-categorized series of steps that it follows in order to stop infections and get rid of infections. We're going to talk about that series of steps now because it has to do both with why you get sick and how we're going to vaccinate you from this disease. All right, so let's talk about it. We're going to actually look at these 3 infected cells here and talk about what happens to them. The first thing is that as these cells are replicating the virus and becoming damaged, they actually kind of sense that there's something wrong and start to secrete what I'm just going to call today help signals or cytokines out of the surface of the cells. So all represented by these little colored shapes here. And these cytokines are sort of floating, or these help signals are kind of floating around in the area around the cell. And they start to elicit or start to recruit an early immune response. And this early immune response is made up of a lot of different types of cells, including these cells called macrophages. Okay, and these macrophages actually have a couple of superpowers to them. One of the things that they can do is they can sort of cruise around, and when they find a virus that's just hanging out in the fluid around cells, it can actually encompass them and digest it up into little parts so that it's no longer infectious, right? So these macrophages are going to move around and they're going to find viruses and they're going to kind of eat them up and digest them so that they're no longer infective, thereby kind of slowing down the spread of the disease. The macrophages on their own are not enough to stop the disease, but they're enough to really slow it down so that other cells can get in there and help. The other thing that these macrophages do is that they'll actually secrete more of the help signal that we talked about. So they're going to secrete more of these molecules out into the environment around. And these help signals, together with the macrophages and the damaged cells, are actually going to lead to a bunch of inflammation in the area. Okay, and this inflammation is going to lead to a lot of fluid and a lot of immune cells coming to the site of infection so that they can all start to help with this infection. Now, this early response is taking place kind of right away as you're being infected and as this infection is getting established. And it lasts, or it starts to kind of establish itself, and it lasts all the way through the rest of the infection. So working to clear this infection, stop the spread of the infection. And it has a really important role also in recruiting sort of the second stage of the infection, what I like to call the late response, or that immunologists call the adaptive response. And this late response is what's actually going to clear this infection. It's going to get rid of the viruses that are outside the cells, and it's going to kill these cells that are infected. Okay. A couple things about this late response. First of all, this late response takes somewhere between 10 and 14 days to really get established. And that's why when you get sick, you kind of feel sick for 10 to 14 days while your immune system is really getting rid of what's inside you. It's also why when you get vaccinated, you're not fully vaccinated until 10 to 14 days after you get your shot. Okay. The other thing is that this response is actually a combination between 2 cells that we're going to talk about a lot more here in a second, called B cells and cytotoxic T cells. Okay, and these two cells have important roles in stopping the infection, but they also establish something called immunological memory. And this memory response is what prevents you from getting sick if you're ever exposed to this virus again, or if you're exposed in close time to this virus again. It's this immunological memory that we're going to talk a lot about when we talk about the vaccines. Okay. So I want to talk a lot more about this adaptive response because it is the kind of fundamental foundation of vaccines. But before we do that, I want to stop here and talk about what happens when you get sick from an infection with SARS-CoV-2. Because at this point, all we know is that the virus is in your body, but we haven't really explained right now why it causes all of those respiratory symptoms and the other symptoms in your body. So a couple things here just in terms of terminology. I told you the virus is named SARS-CoV-2, but the disease is called COVID-19. All right. So if you have SARS-CoV-2 in your body, if you're infected, you don't necessarily have the disease. In fact, about 40% of infections with SARS-CoV-2 lead to no symptoms. They're completely asymptomatic. But if you show symptoms, then you have COVID-19. And COVID-19 really comes about because of the time or because of an imbalance in this early response and the time between the early response and the late response. Okay. So here's the idea. We said that we have this phagocytosis, we have more help signal being generated, and we have a lot of inflammation in this early response. These help signals actually can actually cause a fever. And this is actually a helpful response in your body. Raising the body temperature actually decreases the efficiency with which the virus replicates. So it's actually something that's slowing down the infection. Too much fever is a bad thing though. So when this help signal gets out of control and you have too high of a fever, it can actually cause damage. Inflammation is the same. We talked about how important inflammation is to generate this late response to get rid of the virus, but too much inflammation can cause a big problem. Think about where we're talking about here. Most of these infections are taking place in the airways, right? If we have inflammation in the airways, we're actually constricting the tubes that air can get into your body, and that's going to make it harder to breathe. Okay, so a little bit of inflammation is great. Too much inflammation causes big problems. As part of this inflammation, there's also the accumulation of fluid in your tissues. When fluid is accumulating, especially deep in your lungs, it makes it really hard for oxygen to get from your lungs into your blood. And this is what starts to cause shortness of breath and low oxygen saturation in your blood. And the combination of this inflammation and fluid accumulation in severe cases can actually cause somebody to be put on mechanical ventilation to help them breathe. Okay. All of this, together with the virus infecting and the activity of the macrophages, is also causing tissue damage. And when that tissue damage is happening in your lungs, it's causing scar tissue to form, it's reducing the elasticity of your lungs, which makes it harder to expand and contract your lungs, leading to all of these types of breathing problems we see with a traditional severe COVID-19 case. Okay. And when this response gets really out of control, it can actually lead to the formation of blood clots. And these blood clots can form in any tissue that can be infected by the virus, including in the lungs, in the kidney, in the cardiovascular system, in the heart, and even in the brain. And these blood clots can cause all sorts of damage as well. All right. So the idea is that this kind of early response is really important in informing this late response. But if we have too much of this early response, it can actually tip us over into disease itself. So I told you before that 40% of infections with SARS-CoV-2 are completely asymptomatic. And that's sort of the model case that we would have, right? This is how we want our immune system to work. The early response comes in, it creates just enough inflammation to activate those B cells and T cells so that we get that late response. You don't ever feel really sick. Maybe you're a little tired, but you don't ever feel sick, don't really have a cough, any of that, right? But if that early response gets too extensive, that's when we start to see symptoms. A little bit too extensive, you start to have a heavy cough. A little bit more, you start to have a heavy cough, reduced oxygen saturation. A little bit more, you start to get that tissue damage. A little bit more, you start to see the accumulation of those blood clots. If that response gets too extensive, it can lead to death. Okay, so it is a range of response across asymptomatic infections to the most severe COVID-19 cases. And one of the major avenues of investigation into COVID-19 right now is to figure out what tips the balance. And we know that there are some pre-existing conditions that can really tip the balance over towards inflammation and over towards this extensive inflammatory response. Things like obesity, type 2 diabetes, heart disease, and other kind of chronic conditions can actually help to tip the balance towards that more severe case. All right. Excellent. So I want to stop talking about the disease now and go back into talking a little bit more extensively about this late response, because this late response is what we're super excited about when it comes to talking about vaccines. Okay. So I'm going to just go back to these two cells just to simplify the slide a little bit. Remember that when a virus gets into a cell, it can actually replicate itself in the cell and escape from the cell so that it can then go on to infect other cells. So this adaptive response has to actually deal with virus in two different locations. Okay. First, it has to deal with virus that's escaped from a cell already. So it's replicated and escaped. So we actually have virus that is outside of the cell, right? But we also have cells that are infected and that have virus inside of them. So when we talk about the adaptive response, this late response takes 10 to 14 days to generate. We actually need two different tools. One to help us deal with the virus that's outside the cell, and one that's there to help us deal with the virus that's inside the cell. Okay, so we're going to talk about those in turn. We'll start with the virus that's outside the cell. Just a reminder to you, a virus that's outside of the cell is actually super dangerous because if the spike protein connects with an ACE2 protein on the surface of a cell, it can enter the cell and start to replicate. Okay. So during that 10 to 14 days that we talked about, there's actually a activation of a specific type of cell called a B cell. Okay, so in that 10 to 14 days, the B cells are activated and they replicate and they start to make these proteins that are called antibodies. And antibodies are really just proteins that are shaped like a Y, and they have two ways to attach to the virus. So at the ends of both of the arms of the Y, they can attach to the virus. And the antibodies that tend to be the most useful are the antibodies attached directly to the spike protein. Okay, and they attach to the spike protein, and when they do that, they get in the way of this spike protein interacting with ACE2. If this spike protein can't interact with ACE2, it can't infect the cell. So over those 10 to 14 days, as the B cells are making all of these antibodies, they're building enough antibodies so that when viruses enter the cell or enter your body, the antibodies can bind to all of the spike proteins and get in the way of any of them interacting with ACE2, which means that now this virus can't get inside the cell, it can't replicate, it can't escape the cell, and it can't infect others. Okay. So when we are talking about viruses outside the cell, it's really dealt with by the adaptive response, the B cells that make antibodies. And these antibodies specifically are binding to the spike protein on the surface of the virus. Okay. All right. So that's one, right? We got rid of the virus, or we have a way to start to conquer the viruses on the outside of the cell. But we also have viruses inside the cell. So if you remember, we have these airway cells infected with the virus. These cells are not completely helpless. We talked about the fact that they're already secreting help signals, but they also do one other really helpful thing, and that is that they actually take little pieces of viral proteins and they put them on the surface of the cell. So for example, they can take little pieces of the spike protein of the virus and put them on the surface of the cell. And this acts as a notification to the adaptive immune response that this cell is infected, that there's virus inside here, and that the immune system needs to take care of the cell, needs to get rid of it. Okay? So these spike proteins on the surface of the cell, over the 7 to 10 days, activate a whole army of these cells that are called cytotoxic T cells. Okay? Cyto means cell, so these are toxic to cells. And these cytotoxic T cells have a little receptor on them that can bind specifically to that spike protein. And when it binds to the spike protein, it actually releases a whole bunch of poison onto the surface of this airway cell. Okay, this poison is a special poison that causes the airway cell to kill itself. And it kills itself in a way that actually keeps all of the virus contained inside it so that none of this virus can escape and go off to infect other cells. All right, so the cytotoxic T cell kills any cell that's infected with the virus, and it does it by recognizing the spike protein on the surface of this airway cell. Okay, so taken together, the B cells that make the antibody can protect from the virus that's outside by binding to the spike proteins and preventing it from infecting. And if a virus happens to make it inside a cell, that cell will signal to the cytotoxic T cells, and the cytotoxic T cells will kill it. So we're able to take care of the virus in the two places we find it, outside the cell and inside the cells. As part of this response, in addition, we also make cells that are called memory cytotoxic T cells, or memory B cells. B cells are long-lived cells that will stick around in your body for months after you've been infected with the virus. And these cells are continuing to make more antibody and secrete it into your bodily fluids. And the cytotoxic T cells are continuing to circulate around and look for any cells that might be infected with SARS-CoV-2. Okay, this gives you protection for months after you've seen the virus. If you're exposed again, before the virus can get in and really start to replicate and infect other cells, the antibodies take care of it, the cytotoxic T cells take care of it. Likely you don't even feel any symptoms at all from this infection. Okay. All right. One more reminder before we move on from this, and that is that both the B cells and the cytotoxic T cells that we're making memory cells out of are responding to the spike protein in a natural infection like this. So when the whole virus comes in, the B cells and the T cells are responding to the spike protein. Okay. So understanding this immune response and understanding that both of these cells are responding to the spike protein actually gives us a way to manipulate the immune response to create immunological memory without ever having to take the risk of being infected by the virus itself, right? So the whole idea of a vaccine is to generate this immunological memory without having to take the risk of the low oxygen saturation, the scarring in the lungs, the blood clots accumulating in your heart and in your kidney and in your brain. Right? If we can generate that memory, we can protect you so that then if you're ever exposed again, you won't get sick. So the idea here is that we want to somehow get spike proteins into our cell, all right? And then we want to activate the B cells and the T cells to make memory cells so that if we're ever exposed again, we're protected. There are a couple of different ways to think about how we get the spike proteins into our cell. The first that I often hear people say is that, can we just vaccinate directly with spike proteins? Can I just put a shot right in my arm that's got spike proteins in it that will activate these cells? And the answer to that question is probably yes. There are companies that are working on that. There's a company here in the US called Novavax that is working on a protein-based vaccine. And we have other vaccines for other diseases that work like this. The problem is that we don't generally get a lot of proteins into those cells.
Hey, Sarah, there's a question someone's posted that I thought I might interrupt. It's a kind of timely question. So it says, should someone who has already recovered from the virus, which means they have developed antibodies, still get the vaccine?
Yeah. So the recommendation is that, yes, you would still get the vaccine. And the reason for that is that every time you're exposed to the virus, or the vaccine in this case, your response gets more robust and stronger and more reliable. So having exposure again through the vaccine just boosts your response so that you're more protected in the future. The second reason that they're suggesting people do that is because at this point, we only have data that says that your immune response, these memory cells, lasts for about 6 months. I suspect it's going to be longer, but right now we only have the data that says 6 months. And so the idea is that you want to get vaccinated before that 6 months gets up so that you can continue to have the protection. Okay.
There's another really interesting question, which says, if the memory cells protect on the long run, then why the booster dose, and especially with the Pfizer vaccine?
Yeah, so the memory cells do protect, and actually you'll notice that there are vaccines like the Johnson & Johnson vaccine, for example, that you only need 1 dose. You may have heard that the Pfizer vaccine and the Mrna vaccine actually give you about 80% protection after the first dose, right? But what happens is that the next time that you see the vaccine or the virus, your response gets even bigger and you make more memory cells, more antibodies, and more cytotoxic T cells. And all of that means that you are more protected the next time you see it. And that's why you see the numbers change. So for the first Pfizer dose, you get about 80% protection, right? The second Pfizer dose, you get about 95% protection. And that's just because you amplify that response during that second dose. And it's not uncommon to have multiple doses of a vaccine. So that's actually very common in both childhood vaccines and vaccines that we give adults, that you need multiple doses in order to have a robust enough immune response that it protects you in the future.
Thanks, Sarah. There's another question, but I think it's more about the vaccine itself. And so we're going to wait. Let's wait for a few more minutes from you and then.
Yeah, actually, I'm going to answer this question real quick. I can see it in the chat.
Okay.
So it's basically saying, what's the difference between cellular and humoral immunity? So cellular immunity is this, the cytotoxic T cell. Humoral immunity is the antibody response. So that's the difference right there. Is humoral is the antibodies, cytotoxic T cell is cellular. Cellular immunity is not necessarily stronger, but it seems to be something that lasts a little bit longer for this particular vaccine and this particular virus. So it's not necessarily stronger, it just seems to last a little bit longer. But think about how powerful that is. I mean, it's killing cells that are manufacturing and infected with the virus. So it is a very powerful response. All right. I am going to talk about J&J vaccine and the 3 vaccines here in a few minutes, but I'm going to go ahead and finish up talking because I do want to get you the information about the vaccines that we're giving here in the US.
Okay.
Cool. All right, so we could just vaccinate with spike proteins, but that's not actually super effective as of yet. They are working on that. One of the more classic ways to vaccinate is actually to think about how we can get the cell to actually make proteins itself. And there are a lot of ways we've done this classically, but in the last couple of years and with the vaccines we're working on now, what we do is we vaccinate with genetic information so the cell itself makes its own spike proteins, okay? And we know we can do this because we know that cells are professionals at making proteins. Every cell makes proteins, and every cell makes proteins in the same way, all right? They use information from the DNA, which is like the instructions on how to build stuff, to make a molecule, a very temporary molecule in your cell called an mRNA. And then that mRNA is used by a structure in your cell called a ribosome that builds proteins, okay? It's a super efficient system, but it's not very picky. So if any mRNA shows up in the cell, the ribosome is going to make proteins. Right? So if I can get an mRNA in the cell for the spike protein, I can just let the cell itself make the spike proteins. And that's really what's happened with both of the vaccines that are being used here in the US. And we will talk a little bit more about Johnson & Johnson if you guys have questions about it here in a second. But the Pfizer and Mrna vaccines are both what are called mRNA vaccines. All they are is a tiny piece of mRNA that has only the instructions for building the spike protein. And it's wrapped in a little bubble of lipids that allows it to get in the cell. And as soon as it gets in the cell sees it and it's like, oh, mRNA, I gotta make the protein. And the ribosomes make the protein and end up making a whole bunch of spike proteins and putting them on the surface, activating both the B cells to make antibodies and the cytotoxic T cells to go off and monitor for cells that are infected with the virus, okay? Super effective. The Johnson & Johnson vaccine is actually what's called an adenovirus vaccine. It's an empty virus. So it's a virus that we know can infect human cells, but everything's been taken out of it except for a tiny piece of DNA that they've put into it that has the instructions to build the spike protein. So just like another virus, this cell attaches to the surface, or this virus attaches to the surface, it gets into the cell, and the DNA then instructs the cell to make an mRNA, and the mRNA instructs the cell to make the spike protein. Okay, so we've basically taken what we know about the cell and used it to our own advantage to make a bunch of the spike proteins so that we can activate the B cells to make antibodies and the cytotoxic T cells as well. Okay, and these vaccines actually work really well. So we have the Pfizer and Mrna vaccines both have efficacy, which just really is about effectiveness rates of about 95%. And Johnson & Johnson has an efficacy of about 72%. And I should say that these were calculated a little bit differently. If you're looking just at severe COVID-19 symptoms, this number is much closer to 95%, just like these vaccines here. So the Johnson & Johnson vaccine does a great job of protecting you from severe disease, that disease that puts you in the hospital, gets you on a ventilator, causes all the blood clots, could potentially kill you. Okay. So really effective vaccines. I see other questions, so I'm just going to go ahead and address those questions. I have someone asking, why are there not second doses for the Johnson & Johnson vaccine? Part of that, so it's interesting. I suspect that eventually there might actually be a second dose of the Johnson & Johnson vaccine. The goal right now is to get as many people vaccinated as possible. And the adenovirus vaccine is actually something that's really easy to transport and to get to hard to reach places. And so the idea with the Johnson & Johnson vaccine is that if we could have a one-shot vaccine, we could get more and more and more people vaccinated as quickly as possible. But as we start kind of talking about variants and the virus changing and so forth, I suspect that if and when we start giving the Johnson & Johnson vaccine again, that we could expect probably in 6 months to a year that we'll see a booster shot, which might actually start to cover the variants as well.
And Rebecca is asking a sort of related question, I would say. She says there are 3 vaccines. Should students wait to get one they want, or just get vaccinated ASAP?
Yeah, so the traditional, so the best advice that I have is to get vaccinated as soon as you can. The whole purpose of vaccine, I mean, there's lots of purposes of vaccines, but one of the major ones is to prevent you from getting seriously ill. And all of these vaccines do this with almost the same efficiency. And so getting vaccinated is a really important part to protect yourself. The other thing that's really important about these vaccines is that the more people who are protected means less people who are transmitting the virus. We were going to talk a little bit about this concept of herd immunity. In fact, let me see, I can just show you this slide. The idea with herd immunity and with more people getting vaccinated is that the more people that are immune, the slower the virus spreads. And this picture actually does a really good job of kind of talking about that. So if all of these little purple people are susceptible, which just means that they can still get sick, and one person gets sick in this community of purple susceptible people, the virus can actually spread really quickly and really efficiently. And this is what we saw happening in the big surges that we had in this country, where we had communities getting sick, and you would see little clusters of the virus spread very quickly because people didn't have any immunity. If we can generate immunity by vaccinating people so that they can't get infected, it actually protects the spread, it stops the spread or slows down the spread. The idea being that if somebody gets infected and they're surrounded by immune people because they were vaccinated, it doesn't spread. And even when it does spread, it can't spread far because there's so many vaccinated people around. So you may have heard this concept of herd immunity, like when are we going to reach herd immunity? And that would be an ideal goal where we have so many people vaccinated that even if somebody got infected, it couldn't spread at all. We're really far away from that. And honestly, it's going to be hard to get there, especially when we're not vaccinating kids yet. But it's not an on-off switch, this idea of herd immunity. The more people that are vaccinated, the less the virus spreads. right? So even if we can't get all the way to the 75% herd immunity that kind of immunologists are touting, even if we could get to 50%, that means that there are less vectors and less reservoirs for the virus and less people will get sick. So I keep telling I have a daughter who's 15 years old and she's desperate to get vaccinated so that she can go back to her 15-year-old life. And I keep telling her that she can't get vaccinated. It's just not, the vaccine's not approved for her. But the more people around her that get vaccinated, the more protected she is, and the more likely it is that she's going to be able to go back to her life anyway. So even if she can't get a vaccine, if we can get enough people around her vaccinated, then she's going to be safe going off to do what she wants to do, whatever a 15-year-old does.
A sort of related question is, at the moment, children are not getting vaccinated. When do you think it'll be available for children?
Yeah, so I'm keeping a close eye on this because my daughter is asking me to. They have already done so Pfizer has already done a trial and submitted results for kids from 12 to 16 years old to the FDA. I suspect there's going to be emergency use approval for that sometime in the next month or so. How fast they start rolling it out to those kids is another question, but I expect it will be approved really quickly. The results were amazing. Kids have an even stronger antibody response than adults do. Or these young teenagers do, I guess, have an even stronger antibody response than adults do. Both Pfizer and Moderna have also started trials in kids who are younger than 12. And they're doing right now, I can't remember the age ranges, but it's something like 9 to 12. And then they're doing a group in a couple months. They're going to start the next group below that. And in a couple months, they're going to start babies 6 months up to I think it's 5 and then 5 to 9. Anyway, they're in the process of doing that. I suspect for young teenagers, it will be soon. They're hoping that by fall for school age kids, but we'll see how fast that goes.
There's a question clearly from a biologist that says, is the spike protein from the mRNA vaccine secreted or membrane bound, or both?
It's both. Yeah, it's both. That's a good question. So it's both. And actually, the fact that it's secreted makes it easier to activate the B cells. And the fact that it's membrane bound makes it easier to activate the T cells.
Got it. There's one question that talks about rumors that there are fetuses in the injections. Is this true or false?
Yeah, so like a lot of the kind of misconceptions around vaccines, there's kind of a root of truth in this, sort of. And here's the deal. There have been vaccines in the past that have been made using fetal cells or fetal cell lines. So not even taken from fetuses, but cell lines that have been passaged many, many times. The vaccines that we're talking about, the Pfizer and Moderna vaccines, are not made in that way, there are no fetuses, there are no babies being killed to make these vaccines. They are vaccines that have been passaged through research, and there are fetal cell lines used in the research that has come to them, but they themselves don't contain any direct fetuses, no.
Got it. Thank you. I don't know if you wanted to continue, because there's questions about side effects and symptoms and so on. So
I'm happy to just keep answering questions. I have some stuff about side effects if you want to see it. Let me see if I can find the slide. Is side effects the next question?
Yeah, so the question says, is the fact that the J&J vaccine is part of an adenovirus the reason people get stronger symptoms than Pfizer or Moderna?
Yeah, so the Johnson & Johnson vaccine actually seems to have milder side effects than the Pfizer and Moderna vaccine. And there's a couple of reasons for that. One is, first of all, it's one dose, and the place that we see most of the side effects for Pfizer and Moderna is in the second dose. So it might actually have to do kind of partially with the fact that there's only one dose. In terms of the side effects, maybe actually what you're talking about is the blood clots, which is something that's come about. I can talk a little bit more about the blood clots. What's been happening with the Johnson & Johnson vaccine, let me see if I can find it. So what's been happening with the Johnson & Johnson vaccine is it went through a normal clinical trial The clinical trial actually had almost 44,000 people in it. Didn't really see any big side effects, safety side effects. But as we're doing with all of the vaccines, including the Pfizer and Moderna ones, we have continued to monitor people who have gotten that vaccine, looking for safety issues that may come up in numbers that we wouldn't have seen in this big trial that they did here. At this point, about 6.8 million doses of that vaccine have been given, so close to 7 million doses. And you may have heard in the news that, I think it was on Friday, there was a pause put on, or maybe it's Thursday, there was a pause put on giving the Johnson & Johnson vaccine because there have been 6 cases of blood clots that have occurred. And these are kind of funky blood clots. They're not traditional, what you would traditionally think of as clotting issues. That have come about in women, younger women who have gotten this vaccine sometime between 1 and 2 weeks after they've been vaccinated. The FDA has actually stopped, and the CDC has stopped giving this vaccine. So we're not giving it in the United States anymore. And they're doing this for a couple of reasons. One is so that they can further investigate what's happening with these blood clots and figure out a way to treat them so that we can monitor for them and treat people for them so that they don't die, because one of these people actually did die. But it's a clotting issue that if we know is happening, we can actually stop. We just weren't aware that it was happening. And so this pause is kind of for us to kind of get all of our ducks in a row and make sure that everybody's safe getting the vaccines. You might see that when this vaccine is when we resume giving this vaccine again, that we're not giving it to this highest risk category of people, potentially is what I expect will happen. The other thing that I want to just say quickly about this is that while this is a very serious side effect, it is a very, very rare side effect. We're talking, this number might go up because we see these results coming, or we see this clotting problem happen 1 to 2 weeks after vaccination. So while we wait for that 1 to 2 weeks, we might see this number go up. But 6 cases in 7 million, that is a very low risk for these blood clots. And your risk of getting COVID-19 and even getting serious COVID-19 is actually quite a bit higher than that, especially when it's spreading really quickly in the communities. So it'll be interesting to see. I think this gives me so much faith in our system, though, that we were able to quickly get vaccines on the market, and that we stopped 6 cases out of 7 million of blood clots, which is something that we see happening in people all the time, that we were able to kind of potentially look and see that there's causality here and stop and pause and say, what is the cause and how can we stop it and how can we protect people against it before we continue on. This is a pretty amazing monitoring system that we have set up that's caught that, and that is protecting people from it.
Thanks, Sarah. Rebecca also pointed out that the J&J is doing a double dose trial. So that's maybe a second dose of that vaccine may come up in the future.
Yeah. And actually, I'll say also that both Pfizer and Moderna are looking at a booster shot potentially coming out in the fall or early winter that will cover, so will be one more shot that you'll get even if you've gotten the 2-dose sequence, that will also cover the variants that we're starting to see. So I suspect that we're all in line for another shot somewhere along the way.
Excellent. And Lene's question is a little bit more about how vaccines might be made. So she says, I hear people say that vaccine is like putting poison in your body. What would you say to these people? I already took the poison. I just needed an educated response to this.
Yeah. So a couple of things about this. One of the things that I would say is that you are definitely putting something into your body. There's no 2 ways around that, right? You're getting a shot. So clearly something's going into your body. But the things that are in the vaccine are things that are not things that will last in your body. mRNA is a super short-lived molecule. In fact, they actually had to stabilize it a little bit so it lasted long enough to be effective as a vaccine. mRNA is gone within a couple of weeks, and your cells are made of the same lipids that are in the vaccine. So it's really not something that you're not used to seeing, but it's still foreign, right? It's still something that your body didn't make. So let me just put it this way, that the idea is there is a really high risk with COVID-19 for long-term effects. We have a lot of people who've gotten really, really sick, been in the hospital, been on ventilators, will have long hauler syndrome where they have fatigue and cardiovascular problems and lung problems for months on end, if not years after this is over. And all of this is because you have the virus in your system and it's replicating so much and you can't control this response. Whereas the vaccines are a way that we can actually control the response and we can get that protection so that you don't have any of these other issues, right? We all put stuff in our bodies all the time. The food that we eat, the drinks that we drink, the icy margaritas from Friday night, right? We're always putting stuff into our bodies. But this is something that we can put into our bodies that's actually protecting us from something that can be really, really devastating. And even if it's not devastating to you, it could be devastating to you economically. If you get sick and you have to take 2 weeks off from work or 3 weeks off from work to recover, right? It may be that you get sick and you get somebody near you sick, you get your mom sick or your grandma sick or something like that. And so this is about protecting yourself from these symptoms. It is a way for you to do it, and it's a way for you to do it safely. At this point, there's been more than 250 million doses just in the US of these vaccines given. Actually, wait, let me check that number. Oh, I'm sorry, 209 million doses of vaccines given here in this country. And out of these, we've come up with some problems, right? We see the blood clots in 6 people. There have been cases of anaphylactic shock that's happened in a few people right after they've gotten vaccinated. We've seen some problems come up, but 209 million people is a lot of people. And we are monitoring really closely to see what kind of side effects come up. It's just so much safer to get the vaccine than it is to get COVID-19.
Thank you, Sarah. I wanted to also share with you, we're coming up close to the end of our hour. Our Foothill Dianza trustee Pearl Chang says this forum is excellent. Thanks to the team and today's speaker, Dr. Cooper. Very informative and it's a great service. So yes, I do. That's good feedback to hear. There's a question. Moderna and Pfizer are both working on boosters to be taken in a year or so for extended immunity, as well as for the further protection against the new variants. If only one of them completes the booster due to unforeseen issues, would someone who received the Pfizer be able to get the Moderna booster and vice versa? Ooh, that's an interesting question.
Yeah, it's an interesting question. And actually, I don't know what the policy on that would be, but I suspect that it would be that's exactly what would happen. It's really interesting, actually. In our country, we've been really faithful to the vaccine manufacturers' instructions and how they did their trials. If you've gotten your vaccine already, you know that you go in and you schedule your next appointment for 3 weeks after if you got Pfizer or 4 weeks after if you got Moderna. But in other countries, they're actually doing a lot of different things to try and get as many people vaccinated as possible, including giving people 2 shots, whatever they happen to be in whatever order they happen to be available. And so that data is coming about whether it's safe, if it's safe to have, or if it's effective to have increased or decreased frequency between the vaccines. And by fall, when all of this is happening, we're going to have all that data at our fingertips, and we're going to be able to look at it and see whether it's safe. And I am 100% confident that our government's going to make sure that we have the booster, whatever we need.
There's one more question, which I think is really interesting. We've all been told that if you're reacting to the second vaccine, that's because that means that the vaccine is working and it's doing its job. But should someone be worried if they don't get any symptoms after the second vaccine? Is it still working? Are we generating antibodies, or nothing's happening there?
I'll tell you a really funny story. I've heard that, and I know that, right? The reason that you get those symptoms is because you have this response happening in your body, right? You have these help signals being issued. You have the fever, the inflammation. It hurts a little bit. It's happening in your arms instead of in your lungs, which is great, right? But when I got my vaccine, I actually told my husband on the way out the door, I'm like, I really want to feel some symptoms just so I know this sucker is working. And then I felt the symptoms and I was like, yeah, I wish I hadn't felt the symptoms. But yeah, the fact that your immune response, remember we talked about asymptomatic versus symptomatic infections, and asymptomatic infections seem to be just as effective in creating the B cells and the antibodies and the cytotoxic T cells. So you do not need to feel symptoms in order to get the immunity. It is still quite effective if you don't feel those symptoms. As much as we all want an indication that it's working, it is working.
If you want to take a couple minutes to wrap up the presentation, then yeah, that'll be.
Yeah. I'm actually happy to, I'm not all out of order here anyway, but I'm actually more than happy to stick around and answer questions as long as people have them. I think that's the most effective way.
Okay.
We'll take a look at the rest of the prepared slides. I can maybe give them to Sophia and she can post them.
We can post them along with our along with the recording of the entire session. So that was actually one of the questions as well, which whether people will be able to see the rest of the slides that you have prepared. Yes.
Yeah, I mean, the slides, as you can see, I don't actually often put a lot of stuff on the slides, but I do have other slides and I'm happy to put them up.
And one other question that comes up, I think this comes up a lot, is whether the vaccine will protect from mutants.
The variants, yeah.
Yeah, the variants.
Yeah, so the variants, it's actually really interesting. The variants, I had a picture. Let me see if I can find it. The variants, so this is all sciencey, so don't panic. I'm just going to tell you what it is. This is a model of the spike protein, and it's the surface of the spike protein that binds to ACE2. So if this is ACE2 and this is the spike protein that binds right here, and so you're looking, this face right here is what you're looking at in this picture here. And the variants, a lot of the mutations we're seeing are in this surface of the spike protein that binds to the ACE2. And you can imagine that it would be really effective if these antibodies were able to bind in this area, right? Because then they get in the way of those two things binding together. And so when these changes happen, especially this, the most work has been done on this, they call it the EEC variant right here. When these changes happen, they basically are happening in places where antibodies are really great binders. So it's making it so this antibody that used to be able to bind and get in the way all of a sudden can't. So the vaccines that we have right now are still effective against the variants, because even though the antibody might not be able to bind here, I still have other antibodies that can bind in other places and protect me from getting infected. The worry is that as more and more of these variants, more and more of these mutations develop, that they're going to get in the way of other antibodies binding too, and we're going to lose this antibody protection, right? These antibodies are no longer going to be able to bind. And so the reason that we're pushing so hard to get people vaccinated as quickly as possible is not just to make sure people are protected, but also because the reason we see these variants developing is because we have so many people infected, right? These are super rare mutation events, but because there are so many people in the world that have the infection, these super rare events are happening really often. So the more people we can get protected with the vaccine, right now that still protects against all these variants, the less people are going to be around to help brew new variants, and then we might not need more vaccines. Does that make sense? So we're basically saying, if we can get enough people vaccinated now, we won't get new variants, and then we won't have to create vaccines against them. Okay.
Yeah, that makes sense. So, and a couple other questions here are super interesting and that seem to come up a lot. One is, are there people that shouldn't be vaccinated? We've talked about immune compromised individuals and how this could affect them.
Yeah, so for sure, I am not a physician. So if you have concerns about whether you should be vaccinated or not, you should definitely be talking to your physician. But in the grand scheme of things, this mRNA vaccination seems to be super effective, even, or not super effective. It seems to be less effective, but still effective even in people who are immunocompromised. The adenovirus vaccine, the Johnson & Johnson vaccine, is something they probably would not give to somebody who is immunocompromised, but you can still get Pfizer or Moderna. But you need to talk to your doctor if you have specific concerns about who should get vaccinated. But a big population of people can get vaccinated with these, especially the Moderna and the Pfizer one. It's not a virus. It's not replicating. It can't cause you to get sick. It's just something that gets injected into your body and gets broken down a couple days later. So even if your immune system doesn't respond to it, it's not something that's dangerous to you.
And so another question is whether you can still pass on the virus if you've been fully vaccinated.
Yeah, that's the big question, right? That's actually the question we'd all love a really great and clear answer to. So at this point, So there are kind of three things that vaccines, that we would love vaccines to do. The first is to stop you from getting severely sick, which these vaccines all do really, really well. The second would be to stop you from transmitting them, which means that you could still get infected with an asymptomatic infection, but you might be able to pass it on to somebody around you who's not been vaccinated, and then they get a symptomatic infection. We have data on the first. Can it stop a severe infection? And the answer to that is yes. We're starting to get data about the second, about whether or not somebody who's vaccinated can still get an asymptomatic infection and pass it on to somebody else. The data looks really good. It's coming out of a couple of different places. There are some studies that have been done in hospitals with healthcare workers where they're still testing people to see if they have the virus. And there's data coming out of Israel as well that has a pretty high vaccination rate amongst its citizenry, looking to see about asymptomatic infections. And it's looking pretty good. But we don't have the answer to that question yet. And that's why one of the reasons why you should still wear a mask even if you've been vaccinated, because you might still be a risk to infect other people. And you wearing a mask is protecting them. Even though you can't get sick, you might still be able to get other people sick. We're going to get that data really soon. And you're going to have an answer to that question. And we're going to know better when it's appropriate to wear masks and when it's not. But right now, it's important to protect the people around you because we just don't know for sure.
I'm taking over because Ram's internet, of course, just froze. So I'll ask another question. And Sarah, why don't you tell us how much longer you want to go for, and then we can kind of set a time because it's 5:02 now.
Yeah, I totally understand that people would have to leave, and I don't have any problem with that. But I'm also happy to stick around and answer questions while people have them.
Okay. And interpreters too. I know you all are on maybe your own schedule. So whenever you all need to leave, please don't, we are not holding you to that. So Sarah, here's a question. It says, it's the first one on the list that didn't get answered yet. Is it true that if you get a strong reaction from your first Moderna or Pfizer, it probably means that you've had COVID before, as it suggests that your immune system remembers it and mounts an attack right away?
I mean, I don't think we have any evidence to say that or not at this point, but it's not a bad hypothesis, I think. If you're getting a stronger reaction, it indicates that you have some of those memory cells and that your help response is really big. So I don't think it's a bad hypothesis, but I don't know that we have any scientific evidence to say yes or no. At least I don't know any scientific evidence to say yes or no.
I'll hand it back to you, Ram.
Of course, my internet had to go out at the most critical time of this, but anyway, I'm back. There's a sort of interesting question, I think, from Julia, which says, I've read that in Brazil, the infection rate of that variant is growing rapidly among young children. Is that related to the structure of the virus, or is this due to more sociological reasons?
Yeah, it's actually really interesting. Many of the variants are doing better in younger people. So not even just young children, but younger people, kind of that age range, like 18 to 30, that was maybe getting sick or maybe getting the virus before, but wasn't getting very sick, but also amongst school age kids. I don't know that we know the answer to that question yet. We know that these are far more transmissible, which means that it's easier to get infected with them. And so what it might mean is that the threshold for getting sick is just lower, and it lowered down enough that now these kids are able to get infected. And because they haven't been infected yet before, they're just getting infected in bigger numbers. I've seen analysis out there that says that young people are out and about more now. So kids are going back to school. Young people are going on spring break. We have, people are tired of wearing masks, tired of being away from their friends, and they're going out and about. And so the increase in the rate of younger people getting infected might have to do more with behavior. But we just don't know yet the science behind how these particular mutations are really interacting in terms of age. I think there's an argument to go either way.
Great. There's a question very interesting about the storage of the vaccines, which I think let's go to, which talks about the main issues with the vaccine storage outside of the storage conditions. Is it just degradation, or are there any other concerns? And then you've talked earlier about comparing the J&J with Pfizer, Moderna, and the J&J does not require those really cold temperatures and under which it needs to be stored. So it seems kind of relevant to those discussions.
Yeah. So this vaccine, the Pfizer and Mrna vaccine, is actually a really simple structure with this little piece of mRNA. mRNA in your cell is something that's really fragile and very short-lived. It's something that only lasts a matter of hours, maybe a day or so in your cell. And so it's really susceptible to degradation and to breaking down. And the lipid coating that's around it is something that requires really cold temperatures to keep kind of at the solid storage temperature. And so especially these mRNA vaccines really do need to be kept cold. I'm sure they are doing stability studies to see if they can make it so it's not quite so arduous to keep these stored and make it so it's easier to give them. I don't know if you've gotten vaccinated here in Santa Clara, but there's only a couple of sites where you can get vaccinated with the Pfizer vaccine, because that's the one that has the highest threshold for protection storage. The problem with those complicated storage efforts is that it really excludes a large part of the world. Like, there are just not -80 freezers all over the world where we can store these vaccines. And that's the benefit of these other vaccines, like the Johnson & Johnson vaccine or the AstraZeneca vaccine, which is not approved here in the United States, but is also an adenovirus vaccine. Some others that are coming out, Sinovac, some vaccines, Sputnik 5 that's coming out of Russia, they just don't need to be stored that way. And they are one dose, or at least Johnson & Johnson is one dose, which means that it's just much easier to give in places where we don't have cold storage and we don't have the capacity to get people to come back 3 weeks later. When we were talking about the variants just a few minutes ago, we were talking about how important it is to get people vaccinated so that we don't get those variants developing. And the problem is that that's not a US problem, right? These variants are developing all over the world. And they're developing, especially in places where there are high rates of cases and low rates of immunity. And we have to get vaccines to those parts of the world if we're going to stop the development of these variants. And these variants are going to affect us too. The more of these variants that form, the more of these variants that come about, the more they're going to be able to come back to the United States and get us sick, the more booster shots we're going to need, the more lockdowns we're going to have, and that kind of thing. So it's really critical to have these kind of Johnson & Johnson type vaccines that are really easy to give and that are really easy to store. And that is one of the big disadvantages with the Pfizer and the Mrna vaccines. They are great vaccines. They're excellent vaccines, but they're not going to be something that's easy to give all around the world, which is what we're going to need to do in order to stop it, in order to get to a place where we have a little bit less risk from this virus.
Thank you, Sarah. There's two questions that are sort of related, which I'm going to get to together. One says, I heard that someone got the vaccine and then got their blood tested and there were no antibodies. Does that mean that person isn't protected from the virus? And another sort of very related question is, could someone test positive for COVID-19 after they get the vaccine?
Yeah, so I'm actually going to take those in opposite order. So I'm going to take the one, can I test positive for COVID-19 after getting the vaccine? The answer to that question is yes, you can. You can test positive and have an asymptomatic or symptomatic infection. There have been a very small number of breakthrough infections, they're called, where people have been infected with COVID-19 or with SARS-CoV-2. So they've been infected with the virus after they've been vaccinated. Overall, if you look at the numbers, this was just out in the news, just released last week, and I think it was something like 5,800 cases of breakthrough COVID-19 cases out of like 209 million vaccines that we've given. So it's a very small number, but you can still test positive. And so it is important if you feel symptoms still to get tested. Super small number, super small risk, but it is there. And then the answer, or the question about the vaccine and the blood test for the antibodies, there's a lot of things that it depends on. One thing that we know about these vaccines in particular is that the antibody response starts to wane. It starts to kind of decrease as months go on. So especially as we get into 6, 7, 8 months, we see less and less antibodies. So it would depend on if this person was tested, 14 days after their second shot, or if they were tested months later. If they were tested months later, it could just be that waning. If they were tested directly after, this vaccine is effective, but it's 95% effective, which means that for some people it doesn't actually work. And there are some non-responders to vaccines, not just this one, but to other vaccines too. There are non-responders to measles vaccines and so forth that do exist. So it is something that can happen. Again, super low risk of that happening though.
Great. So we do have to sort of wrap up. We're past our time. So I think this has generated a great discussion. So thank you to everybody who attended and asked these wonderful questions. And you can be sure that we will be continuing this conversation for the next 3 weeks on similar topics. So there's that as well. Sara, you wanted to say something?
Just 2 things. I'm looking at questions in the chat that are really important to answer. The first one is, where can I get vaccinated and how much is it? Where you can get vaccinated is on, I don't know if somebody can put into the question and answers.
Yeah, we posted it, yes.
And the second thing I just want to tell everybody that's here and listening, the vaccines are free. You do not have to pay for these vaccines. If you have health insurance, your health insurance might have to pay, but if you don't have health insurance, if you're not a citizen, if you're anything, you can still go and get vaccinated. So make sure that you go to those county sites that are on that list and get yourself signed up for a vaccine. There are very few restrictions about, especially now that it's been opened up to the general public, about who can get vaccinated, and you do not have to pay. And the second question is, can the vaccine itself cause you to test positive for COVID if you're not infected? And it cannot. This is something that's going into your arm. The vaccine or the tests are in your in your spit or in your nose, those are looking for the RNA from the virus, and all you're getting is a really small, tiny little bit of the RNA from the virus. So you will not test positive if you get vaccinated. So if you have requirements at work that you have to get tested and it has to be a negative test in order to come to work, you can still get vaccinated. It will not turn your test positive. Okay, that's all.
Thank you, sir. And a lot of these questions about where to get vaccinated and where to find the vaccine and all of that stuff we'll take up in our future talks, especially starting the next one, which is about public health information surrounding the vaccine. So please do come back and join us. But I want to also end by saying, Sara, thank you so much. And I want to echo some of the sentiments that I've seen in the Q&A and how people have really valued and appreciated all the information that you've been able to share with them. This was super useful. So thank you very, very much.
Absolutely, it's my pleasure. I was glad to be here. It's fun.
And thank you to everybody who was attending this session today. That'll be it, folks, and we will post this recording.
Thanks, everybody.